will be a little bit unusual because it's not a normal standard coherent talk, but more an overview about the different research topics my group is covering. And one of the purposes why I decided to do this in this way is also to show whether they overlap with some of your interests and where we can collaborate together, or whether we can transfer certain things we have developed also to Sunnybrook and other academic hospitals. In addition, I'm doing a lot of 7 Tesla MRI, as some of you know, but I will not talk about it today because I thought it might be more useful when the 70 delivered to 70 to the Sunnybrook is approaching delivery. And so therefore I would be happy to give another talk in 2 years or so in order to talk about 7 Tesla research. But today, therefore, I'm focused on 3 Tesla research. And as I said, it will not be a coherent talk, but more an overview about different topics we are covering. So this is my current lab. It's no particular order. It is a mixture of scientific associate, postdocs, and students from MBP, master and PhD students. Okay, I want to cover 4 different topics. One is MR physics, second one cognitive neuroscience, third in clinical neuroscience, and the last one, which might be of particular interest, is data management. And as I said, this is not a coherent talk. I would be happy to answer questions at any time, and feel free to interrupt me and ask a question without raising your hand. Just unmute and ask the question, please. Okay, the first thing, MR physics. So we are developing a lot of our own sequences, not necessarily develop novel sequences, but adapt some sequences which are already available, but not a product sequence from Siemens, but in order to make it useful for our researchers. And one of the main purposes of the MR physics research, what I'm doing, is to bring sort of advanced MRI to the clinical research. And so therefore topics we are covering are always with the mind that at some point it should be used clinically. The first example I want to show is arterial spin labeling. So as you know, Siemens has an arterial spin labeling product sequence. However, the current implementation of Siemens doesn't include multiband. So what we did is we have actually implemented multiband. And here is an example of blood flow image, 2.5 millimeter isotropic, 64 slices. It takes around 5 minutes, and acceleration factor 2/2. And as you see, we cover actually the entire brain. And as you can also see from the distribution of blood flow in color code, we actually get a very high quality of blood flow all over the brain. So this is, I think, when I talk with clinicians or clinical researchers, they are often reluctant to use blood flow, arterial spin labeling for blood flow or for clinical research. The reason is they have at some point tried it in the past and they didn't find the quality of the images good enough, and they have abandoned that research. But I think now, if you see this kind of images, I think we are actually at this moment quite good with arterial spin labeling, and it takes only 5 minutes, and we get actually very good blood flow distribution over the cortex. We even push the limit a little bit further. We have 1.8 millimeter isotropic, and this takes also around 5 minutes, 5 to 6 minutes. And you see actually still a very good distribution of blood flow in the cortex. And this is, I want to just, for those of you who don't work with arterial spin labeling, usually what people use 3 to 4 millimeter isotropic. So we have, when we use 1.8, we have approximately 8 times resolution compared to what people use in standard, and we have a whole brain coverage. So I think this is really ready for prime time and ready for clinical research. A second sequence.
Sorry, naive question right off the bat here. Those regions that are thresholded out, what's the blood flow there? Is it really that low?
This is sorry, I have not as an overview talk, I have not really sort of put sort of color bar on it, but this is absolute blood flow, and so this is a blood flow in physiological units. So we have acquired additional image to calibrate the blood flow signal. So this, what you are seeing, is actually absolute blood flow.
And the regions that are not orange, you can just see the background. I guess it's
this white matter.
White matter.
Yeah, this white matter. And artificial labeling is notoriously bad for white matter blood flow. So it means it's good for, it has a disadvantage compared to contrast agent approaches like gadolinium, that because the blood takes too long to reach the white matter, the T1 decay already eliminates a lot of the signal. So for white matter blood flow, you would not use artificial labeling. But for gray matter blood flow, as you see in this example, artificial labeling is quite good.
I see. Thank you.
Okay, the second thing what we have implemented is a spin echo grade echo sequence called SAGE sequence. And the reason is we want to do, as you will see later, I'm very interested in not only in artificial labeling for perfusion, but also for dynamic susceptibility contrast using gadolinium. And there are these approaches which are called vessel size imaging, which are combining grade echo and spin echo signals, because they have different sensitivity to blood vessels to extract the vessel diameter, for example, in tumors. So we have, again, this is not our invention, but we have implemented it because there's no product sequence from Siemens. And our implementation has 1, 2, 3, 4 echoes. The first 2 echoes are grade echo, so we can calculate relaxation rates, for example. The third one is hybrid. This can usually be discarded. And the fourth one is a spin echo signal acquired at this echo time. Then the next one is what I was interested in, was wave-KIPI. Wave-KIPI is an approach which was invented by Kâmil Uludağ from MGH, now Stanford. And there he uses sort of a elegant to distribute case-space information evenly, and so that the penalty for acceleration is quite low. And this is a paper from 2019. There they have accelerated 9 times, so 3 by 3, 3 in plane and 3 through slices, and they have acquired space, flare, MPRH, and SWI, and they are all taking around 1 to 2 minutes. And so this is quite impressive that this is how much you can actually reduce the scanning duration and still get good quality. As I said, I'm interested in translating advanced MRI to clinical research, and in clinic, obviously, time is money and health, and to be able to shorten the scanning duration is very important. And wave-KIPI was one of the sequence I was interested. It is complicated because first you have to implement it. We have done this. So Tim in my group is a sequence programmer. He has implemented the wave, which is sort of sinusoidal gradients, which distributes sort of the case-space information. And he has calculated the GMAP, 1 over GMAP. This would be the standard blip KIPI. And the more green, the more signal you lose. Whereas in the wave KIPI, it's sort of the values are around 1, which means we don't lose for 8 times accelerated, we don't lose much signal. We always lose signal because the acquisition duration is less, so we have less data. However, the acceleration itself does not give an additional penalty. So we are working on this. So the sequence is implemented. The problem is at this moment, the image reconstruction is still complicated, and we are implementing our own image reconstruction code in order to reconstruct the images in a good way. Next sequence, what I want to show is, so we are interested again in clinic. Movement is a problem because patients move and we lose a lot of data due to patient movement, or diagnostic quality goes down. So what we are working on is actually acquiring navigators, for example, in TurboFlash, and using these navigator signals to detect the motion and then to correct for motion. Just to give an example, this is a phantom with no motion, phantom with motion, and you see some artifacts here and there. And then if we use radial navigators, we can actually correct some of the motion. It's not perfect, but it's certainly much better than before. We have also implemented this navigator approach for the wave-KIPI. this is now, because at that time we didn't have ethics application approved. We measured on an orange. Here you see the orange with no movement, and then it was then moved manually with a stick. And you see now you cannot see any of these structures. But using this approach, we can actually recover the image quite well. It's not perfect again, but it's this one would be totally useless. This would be still useful for clinical, and the motion was quite excessive. So I think this radial navigator approach will be quite useful for a sequence you choose wave Kaipi or turboflex like MPRH. So we can use this for motion correction. One of my students, Brian, is actually using also trying to correct for motion, but he in all of the data, navigators are either not implemented or they are difficult to implement because of timing constraints. So for this kind of data, we want to use data consistency to correct for motion. So if you have a motion corrupted image, then Haskell has proposed a method with CNN block, and then estimation iteratively the motion and the image reconstruction, then get a motion corrected image. We have modified this approach. I will not go into depth of the modification, and the work is still in progress. But if we, in our implementation, when you look at, for example, motion, this is now simulated data, but we have also experimental data on which we can show the same effect. This is now a corrupted image. You see here, for example, these stripes, which are coming from the motion. And then using this approach, we can actually get quite a clean image. If we have severe motion, for example, this kind of data would be totally useless in clinical environments. If we do this, motion remains. However, we get this sort of from totally useless to somewhat useful. And so this is another approach we are trying to pursue. Now, one thing what might be of interest for you, for Sunnybrook and for others who have scanners, and the scanners are not perfect, so they misbehave. And especially for advanced sequences, this misbehavior of the scanner is a problem. And especially in this case, I'm talking about the gradients. And there's a method developed by the Klaus Prüßmann group. It's called gradient impulse response function, where we are actually experimentally measuring how the gradient behaves. So we have a prescribed trajectory in K-space, and then we actually measure how the gradient is behaving, and we determine the gradient impulse response function, and then use that knowledge to correct the data. And the way it's done, this has been published in several papers, is you acquire apply gradients and measure two slices with positive lifts, with negative lifts, and then calculate the gradient impulse response function. Again, I will not go too much into depth, but this is something what we have measured. And if you do this, you get this kind of curves. I will just slowly walk you through a gradient X, gradient Y, gradient Z. This one is the gradient impulse response function as a frequency, which means how quickly you switch gradients. And then usually we are between -10 and +10 kilohertz. So we want to be in the flat range. And then you have a phase, which then indicates the delay of the gradients. And then we have done two measurements 8 months apart in order to see whether the gradients change, because scanners get older and they might change. And then how does it manifest itself in the K-space data? This is now a zoomed in version of an API trajectory. The blue one is nominal trajectory, and the green one is the measured trajectory. The difference looks quite minimal. In API, also the problems are more controlled. However, this is now zoomed in for spiral. There you see differences between the nominal and the measured gradient impulse response, gradient trajectories. Now, this is quite abstract. For those of you who are not in this field, this might sound very abstract and also quite irrelevant, but I want to show you how it actually plays out in real data. We have developed a pipeline. By the way, this is publicly available. It's in GitHub for image reconstruction it's using the MR Recall in Julia. And some of the steps have been already programmed by other people. And then we can use gradient impulse response function and B0 and other things in order to reconstruct images. But I don't want to go into detail, but if anybody's interested, the code is publicly available. Now we have developed a spiral diffusion sequence because diffusion, being able to reduce the echo time is quite relevant. And therefore, we want to test whether this approach works in spiral diffusion. And then this is work done by Lars Kasper in my group. This is now Phantom. We are now collecting in vivo data. This would be when you use nominal trajectory on a Phantom with sort of these lines. You see here on top, for example, it looks quite weird, whereas here in the middle, it looks quite sharp. If we correct for B0 effects, you see actually a lot of these effects are gone. However, you still see shades of darkness and brightness, for example, here. If we correct for the delay, which we have measured with the gradient impulse response function, actually, this image looks very, very clean. And then if we correct for the full gradient impulse response function, not only for delay, there's some improvement, but the improvements are then a little bit minor compared to previous ones. Okay, how am I with time?
Good. Yeah, you can take up to the hour, but maybe just leave like 10 minutes for questions if you can.
Okay. So here now I want to show something which is not really research, but it might be useful for also for you. We have tested at the Prisma the different coils we have available, the 20-channel coil, 32-channel coil, and 64-channel coil. I don't know what coil is available for you. And these coils have different diameters. So therefore, for bigger heads, usually people use 20 channels, but 64 channel is sort of has more channel count, therefore is superior in certain terms. And we have explicitly tested sort of standard sequences, how they perform. And I will just scroll through some images to show how they look like. This is MP-RAGE. You see 20 channel, 32 channel, and then 64 channel. Then FLAIR, we have acquired 20, 32, 64. SPACE, 20, 32, 64. And they are all 1 millimeter isotropic, and they are standard acquisition duration of 3 to 5 minutes. Then diffusion weighted imaging, again, 20, 32, 64. This is now fractional anisotropy. We have also implemented advanced sequences like NODI and calculating ODI. This is then diffusion the CNR. Again, 20 channel, 32 channel, 64 channel, and you see differences in image quality. Then this is for fMRI, 32 channel and 64. So for fMRI, for example, 64 channel is most superior for resolution of 2.4 millimeter isotropic. I went very quickly through because it was not research, but I want to just share you our experience with it. With structure, when we just do T1, the different coils have comparable performance. And if we go to more advanced protocols like WaveKP, higher channel count might be useful. For diffusion, we found 32 and 64 channels are obviously capable of high acceleration, and they have a CNR benefit over 20 channels. But the FA and the NODI maps, they are comparable. So if we quantify, that's good news. They are relatively independent of the coils. For function imaging, 32 and 64 channel have sort of a TSNR benefit, especially 64. And we can go then push the limits of high acceleration and get higher resolution and shorter TR. Okay, any questions to the MR physics part?
I actually have one question, Kâmil. It's Gary here.
Hi, Gary.
So off the bat, you were showing the ASL. So I'm just curious, what channel coil did you use for that? And then if you were using like a 20 channel coil, if you were to go up to the higher channels like 64, I noticed your acceleration factors were a bit conservative. Would you push your acceleration factors then?
So we have used the 64 channel because for 20 channel acceleration is difficult because we have used 2 by 2. It's still possible with 20 channel, but it's already, you will lose a lot of signal. So for ASL, also you need a good tagging. So there also the 64 channel is actually quite good. So we use, in general, I am more on the conservative side when it comes to acceleration. I don't like to accelerate more than factor 4 because the gain in time when you accelerate more is not that much anymore. You gain, obviously, the scanning duration gets shorter, but the total duration of the scanning includes the pre-scan, the frequency adjustment, and so on. If you add that up, further acceleration does not give you too much gain in time anymore, but you lose signal. So therefore, I usually, I would not go, say, to higher than factor 3. So factor 3 is certainly, multiband is certainly fine, but higher than that, for example, what the Human Connectome project people have done, they have done 8 or even, I think, sometimes 16. That's, I think, going too far. I would go for factor 3, but not more.
Great, thank you.
And the same in plane, factor 3. So therefore, we can accelerate a little bit more than we have done, but I would say the gain is in time is actually not that much anymore.
I see. Okay, thanks.
Any other questions?
Hey, Kâmil, can I ask a question? It's
Yes.
Angus. About your gradient impulse response functions, I played around with these in the past, but I guess in the past I tried to do these for like oblique slices, like oblique like 2D images, and I guess for those, like the cross terms kind of kill you. I guess like with field cameras you can measure those cross terms, but like for like if you don't have a field camera, can you measure those? Like have people tried that, or can you
Yeah, in principle you can measure this. It's not done in the standard way. Usually it's done sort of orthogonal, but there's no reason why you could not do this in oblique slices. You can do this if you want to sort of characterize the gradient impulse function even further, then you can do this. And then it depends on how nonlinear the entire system is. If it's linear, then obviously you would be fine with the without the cross terms. But if it's nonlinear, then you have a problem. Yeah. Yes. So.
Okay.
Okay.
Cool. Thanks. Yeah.
Okay, then I go to the second topic, cognitive neuroscience. I will be a little bit fast on that because a lot of these things are work in progress. So there I want to just show what we're working. So as some of you know, I worked a lot on hemodynamic modeling. So hemodynamic modeling means the fMRI signal, what we are measuring is the bolt response, and we would like to make statements on the neuroactivity. However, there are different steps in between going from neuroactivity to bolt response. So we have to describe the neuroactivity in terms of differential equations. The second one would be the neurovascular coupling, which changes the blood flow. Then we have hemodynamic changes, blood flow, blood volume, and deoxyhemoglobin. And then the MR physics, the sequence, gradient echo, spin echo, then translate these physiological changes, especially deoxyhemoglobin, to the fMRI signal. So this chain of events, there are models, and my group has published several papers on this. And you can do model inversion with this, so which means you measure bolt response and you calculate the neural response most compatible with the measured bolt response. And this kind of model is utilized also in analyzing connectivity between brain areas during cognitive tasks. And this is often used in DCM. DCM stands for dynamic causal modeling, invented by Friston, but the modeling itself is not bound to that. In DCM, there are two different problems currently. One is the effective connectivity is always calculated for a run, which means you have a run where you have a cognitive task, and then you calculate how different brain areas are connected to each other, and then you can compare, say, healthy subjects in patients, let's say a patient with schizophrenia, whether they process the stimulation in the same way as the healthy controlled subjects. However, in more naturalistic stimuli, let's say you watch a movie or you listen to music, the connectivity between brain areas changes the whole time. So we actually need a dynamic effective connectivity and not effective connectivity for a run. The second is because of the model inversion, there are too many possibilities, and therefore, when people analyze this kind of connectivity, the number of brain areas is limited. So usually around 6. But for complex stimuli, there are many more brain areas involved. So Cyan in my group is working on these 2 issues. One is the problem that the dynamic affective connectivity. What he did is just an idea from functional connectivity, which you use resting state, using a sliding window approach, which means you calculate for each window the connectivity parameters between brain areas. And in order to, in contrast to functional connectivity, which just use correlations, we have this physiological model, which is described here. And we calculate for each time point the parameters of this physiological model in using recurrent units. So we calculate, and then we do iteration in recurrent units, and then move to the next time point. And this paper is finished, and we will submit next week. And the results in simulate data are like this. For example, if you have 10 brain areas involved in a task, and you have 2 different inputs, let's say visual stimulation and auditory stimulation, and you have certain connectivity between brain areas, then you get certain bold signal time courses in the different areas, and the color-coded are the fits. And as you see, the fits in model inversion works very well, and we can estimate connectivity parameters very well, which is not shown in this slide, but in the paper we are describing these connectivity profiles. However, one argument might be perhaps it fits every time course very well, and this is not true. So if you would have a different connectivity profile assumed, what you would see, for example, in area number 6, the measured curve is the dotted black line, and the green line would be the fitted curve. And as you see, the fit curve is quite bad, which means this method allows to determine dynamic effective connectivity, but at the same time, it can distinguish between different connectivity profiles in the brain. And we are collecting experimental data using movie watching in order to test whether our approach can extract dynamic connectivity in experimental fMRI data. The next one is number of brain areas are limited, as I told you. And we are using sort of a transformer, which is sort of currently very popular in deep learning literature. I will not go into the architecture too much, but the good thing is once it's trained, the calculation is very fast. And this is what you see in this slide. This is how long it takes to calculate the parameters. The blue one is the neural network aided DCM, determining connectivity, and the red one, the current DCM. As you see, we have a factor 4 to 5 faster calculation of the parameters, which means we can scale up the number of brain areas. And actually, our current implementation is even faster. So I think we are sort of in the process of solving the issue, and perhaps the goal would be to have a whole brain effective connectivity in complex tasks without having the computational limitations. The next topic, as many other people, I'm also interested in AI, and I'm exploring whether AI is useful for fMRI. So what people usually do, they show some images, they have a person MRI, and then you measure fMRI signal. And then the neural network would then do the encoding, so predict from the images the fMRI activation. And most people are interested in the decoding, so measure fMRI activation and reconstruct the images. And this is the current state of the art. So this is the ground truth image, example images shown to the subjects. These are 2 different studies. And as you can see, actually the reconstruction, so this is a fish, and this reconstruction is not too bad. It's obviously not perfect. For example, this donkey, you see the eyes, but it doesn't really look like a donkey, but at the same time, it resembles. So here you see this ski car, and then you have a tractor. So it is good, but it's not perfect. But this is, I think, I would not have expected fMRI is capable of encoding images so well that you can actually do image reconstruction. Tried out is an alternative approach, which is called capsules, which is sort of has been proposed that it's more brain-like. Here's an example of an image, and this would be the fMRI brain activation. Red means high activation, blue means low activation. And one of the CNNs, you see the prediction like this, and you see the prediction resembles, but the scale is a little bit off, but the pattern is actually quite similar. The capsule also looks quite similar in this example. However, if we go to another example where you see a different pattern, the CNNs are doing well, but the capsule is not doing well. So we were quite disappointed by this, which means the capsules which have been proposed to be more brain-like actually do not predict fMRI data very well. Then the next thing, what we invest with AI is called transfer learning, because in these studies where people use to train neural networks, they use 1,200 images, which means a subject had to sit in a scanner and watch 1,200 images. And because the fMRI response is slow, they have to acquire this data over different sessions. So the data acquisition is quite long. Obviously, if you want to do investigating patients, for example, in schizophrenic patients and so on, then you cannot have a patient 10 hours in the scanner or coming 10 times and every time watching some images for 1 hour. So this would not be feasible. So what we asked the question is, can we use actually the data which has been acquired or are available in databases and train the neural network and then use the neural network then on a new subject with much smaller data. So using transfer learning, which means using less data in the next subject. Now, this is sort of the baseline correlation with the predicted fMRI responses. We have sort of 550 test images. So if you have only 100, the correlation coefficient would be quite low. But if you have 1,200 images, the correlation coefficient with the predicted fMRI response actually quite good, 0.5, 0.6, which is quite good because fMRI is noisy. So to have correlation coefficient of 0.5 is actually quite astonishing success. Now, we have evaluated other approaches. Hyperalignment is an approach by Jim Haxby, which doesn't use neural network, but which has been proposed before, how to align different subjects to each other in the functional space. And you get, for example, if you would use, say, only 500, you would get a good correlation. But the best approach, what we have found, are autoencoders. I will not go into the architecture too much, but I want to just show you the result. For example, if we would acquire 300 images in a new subject, we would get better performance than 1,200 images in naive subject. Or to summarize in more plain words, if we have a neural network or autoencoder trained, then if we want to predict the fMRI activation in a new subject, then we need much less data. For example, only 200 images, which would then reduce the scanning duration by a factor of 6, which means we would, instead of having 6 hours of scanning, we would have only 1 hour of scanning in order to characterize the brain activation of a new subject. And obviously, if the more subjects we have, perhaps we can even reduce the number to less. And if people are fine with reduced performance, even 100 images might be sufficient to predict fMRI activation to novel stimuli. Any questions to the cognitive neuroscience part? If not, then I go to the clinical neuroscience. As I told you, a lot of the MR physics, like the motion correction and the wave Kâmil image acceleration, are developed with the view that it will be applied in clinical populations. But in my own projects, I'm very much interested in perfusion imaging and in particular dynamic susceptibility contrast. So in dynamic susceptibility contrast, the first thing what we did is develop a simulation framework. So simulate how a contrast agent changes the MRI signal. So we can have gadolinium usually, but you will see in next slides, we also use deoxyhemoglobin in the lungs as a contrast agent. We have change in arteries. We have then change in the contrast agent concentration in the blood vessels in the local tissue. And then this generates relaxation rate changes. And this all we can calculate with the signal model. The signal model is written here. And this actually from my own paper from 2009, which describes how fMRI signal changes based on blood volume deoxyhemoglobin. And I thought this can also be utilized for gadolinium and other contrast agents. So we have a lot of parameters we can change: blood volume, echo time, contrast agent properties like duration of the contrast agent, blood vessel composition, so how much arteries we have, how much capillaries, hematocrit, and field strength. And experimentally, what we did is we have used the RESPRAG from David Nicholas and Joe Fisher, which is sort of an instrument which changes in the lungs, the blood oxygenation. And then this can be done quite short, like 20 to 30 seconds, which then acts like a bolus going through the tissue, going into the brain and going through the brain voxel. And we have, in this experiment, we have 4 different paradigms. In the lungs, the oxygen saturation in the artery is 98%, and then we drop it to 75% or 84 or 90%, or we start with a lower baseline. Because the drop in blood oxygenation is quite short, this is sort of well tolerated from subjects. So there's no safety aspects, but happy to discuss later. And the reason why we did this is we want to compare whether the dynamic susceptibility contrast calculations of blood volume, blood flow, mean transit time, how much they are dependent on duration, on experimental manipulations. And what we found, these are now experimental data. This is a change of the signal for deoxyhemoglobin, and the maximum change during contrast agent, and this for gadolinium. The scale in gadolinium is higher because the contrast agent, the susceptibility effect for gadolinium is much higher. And what you see is they look quite similar. Obviously, gadolinium is less noisy than deoxyhemoglobin. However, if we build the ratio between these two, what you see if you divide the signal from deoxyhemoglobin by the gadolinium, what you see is actually it's not flat, which means if deoxyhemoglobin would be just a scaled version of the gadolinium, then dividing these two would give a flat map. But what we see is in yellow, these are big vessels surrounding the brain. The gray matter is also different from white matter. So what we have found is actually the deoxyhemoglobin as contrast agent does not act as a scaled version of gadolinium. And actually using then if you go then back to the simulations, we can simulate this and the simulation predict exactly that. Second thing is blood volume as a function of contrast. So blood volume is obviously a physiological parameters. We would hope that blood is independent of whether we use deoxyhemoglobin or gadolinium, or which of the paradigms we are using, which of the 4 paradigms we are using. However, what you see here is the blood volume distribution using gadolinium. And what you see here are the 4 different blood volume distributions calculated for the different paradigms. And what you see is the blood volume is actually varies, the calculated blood volume varies depending on what paradigm we are using. Again, initially we were quite surprised about that. However, this is now, if we calculate the blood volume for the different paradigms, this would be gadolinium, this would be the 4 different deoxyhemoglobin paradigms, ignore the venous part. Then in the simulations, we exactly observe, or we observe very similar pattern. For example, this paradigm is much higher blood volume than this paradigm, which also we saw in the simulation. So even though this is sort of bad news, because this means blood volume calculated with dynamic susceptibility contrast is not reflecting the physiological blood volume. However, the good news is with the simulation, we actually understand why this is the case. And the next project we are working on is actually using this simulation to calibrate the different blood volumes so that regardless what paradigm we are using, we can actually calculate the physiological value of blood volume and not a derived value of blood volume, which might be not accurate. And the same is true for MTT, this mean transit time, this is for gadolinium, and mean transit time, this describes how long the blood needs or the contrast agent needs to go through the local tissue. You would expect that this number is independent of whether it's deoxyriboglobulin or gadolinium. However, this is experimental data. Experimentally, we don't find this. And in the simulations, we also observe that the deoxyriboglobulin predicts much larger mean transit time than gadolinium, which we can confirm experimentally. In addition, even the gadolinium mean transit time is an overestimation to the physiological one. Again, this is bad news because this means we cannot trust mean transit time measurements using dynamic susceptibility contrast. The good news is, I think we are able to understand why this is the case, and therefore we are, in perspective, we are able to correct for it. The next one is next project using by Angelica Manalak is using the grain echo spin echo sequence, which I mentioned initially in the sequence development part, in order to characterize tumor blood vessel distribution. So, and the basic idea is grain echo blood volume is the perfusion of all vessels. And the reason is the extravascular signal becomes for high susceptibility values independent of vessel diameter. Therefore, the grain echo blood volume calculation is the blood volume of all vessels. Whereas the spin echo has a higher sensitivity for smaller vessels due to the rephasing around big vessels. And therefore, it's more weighted, the blood volume we are getting is more weighted towards small vessels. So the idea is then, therefore, if we measure blood volume with spin echo and grain echo, then the ratio of these two or the characteristic of these two gives us an indication whether in the tumor we have more big vessels or we have more small vessels. And this approach is called vessel size imaging. This already is quite an old approach, but it has never made it to mainstream. And we are sort of picking it up. And again, we are using simulations because we can simulate the spin echo signal as we can simulate the grain echo signal. And using the simulations, we can characterize the signal better. This is shown as an example. So similar approaches have been done by the group of Sørensen, published in Nature Medicine. So assume we have in the tissue arteries, capillaries, and veins. And just for illustration, we chose the same size. Obviously, in reality, are not the same size. Then the grain echo spin echo curves would look identical. And if you plot the spin echo as x-axis and grain echo as y-axis, we would get a linear curve. However, if we have overrepresentation of artery, then a grain echo starts a little bit earlier, and spin echo decays a little bit slower to baseline, and then you would get a curve like this, hysteresis. However, if you are more venous dominated, you would get a curve like this. And which means if we measure grain echo and spin echo, plotting their time courses towards each other can give us indication whether, not only whether what the average vessel size is, so the vessel size imaging, but also whether we have what kind of blood vessel composition we have in the tissue. And we have now acquiring experimental data on 2 more patients. My mouse does not move anymore, but I hope people can still hear me. Anyway, my mouse does not move, so therefore I have to just describe the graphs. So these are the 3 different echo times which we have acquired with our custom sequence. In bottom left, you see the normal appearing white matter. You see the different signals. The grain echo signals are echo 1 and echo 2. These are blue ones. And the spin echo signal is the one purple. In tumor, however, you see a very, very different time course. In the first echo, the signal goes below baseline. And this is quite a surprising effect, but the second echo, we don't see this effect. And the reason for this discrepancy is actually leakage into the tissue, which means the discrepancy of the time course between the first and second echo and grain echo gives us information about the leakage. And the spin echo, as you can see, is a little bit wider than the grain echo. So, which means in our model, we have to incorporate now leakage and then correct for it and then do vessel size imaging. Other experimental data, these are now time courses from blood vessels. Again, 3 different echo times, and red is artery and blue is vein. What you see is the characteristics change between the different time courses. If you look, for example, at the echo time 92, this has to be milliseconds, sorry, it's not seconds, milliseconds. What you can see is you see this regular oscillations. This is heartbeat because it's an artery. Whereas in the echo time, first echo time, you see also this heartbeat, but much less pronounced. And in vein, the signal is much delayed, and you don't see this heartbeat-related changes because the deoxyhemoglobin and also the heartbeat dampens the further downstream the blood vessel is. Okay, this was the clinical neuroscience part. Any questions, comments on this? If not, then I will just finish then the last part if I still have time, and that's about data management. Again, this is not necessarily research, but I think it might be of interest for people in Sunnybrook and other places, especially when we think about multi-center studies, and especially when the 7D comes and people from different institutions want to access data. Then we need a data management framework where people are capable of sort of accessing data and having joint analysis pipelines and so on. The XNet, this is a collaboration with CAMH, with the group of Sean Hill. XNet is sort of established, and the version we are using is the one which was developed by them. So you can upload data directly from the scanner. And then you can, it has a folder system which you can organize and share. And you can, for example, when you acquire data, you can, when you log in, you only see your own data. However, if you then want to share data, it's just few mouse clicks in order to enable other users to, if you have a collaborator in a different organization or within the same organization, you just few mouse clicks. And then if the other person gets some permission, and the other person logs in, he or she can then see the data which was then shared with that person. Then you can view and locally download, and the access is very much controlled. And then it is web-based. So that's also a nice thing that you have a web-based interface. And what I'm also very much interested is automatic processing. For example, if there's some common analysis, let's say we want to, fMRI data will always be motion corrected. So if fMRI data is uploaded, the system automatically recognizes fMRI data, does motion correction so that when you log in and download data, you have the original data, which has been unprocessed, but you also can access the motion corrected data, which then saves a lot of time in processing. And some people do not know how to process because that's not their expertise. So certain analysis can also be automatized and brought to the user base. And the XNet abides to the FAIR principle. FAIR means findable, accessible, interoperable, and reusable. Again, the main idea is data management sharing, for example, internally or externally in collaboration. So joint studies or reuse of data. For example, if you collect control data, then we don't have to have control data for each study, but we can share some control data. Obviously, not all control data can be shared, but data is not lost. And in the area of big data and AI, if more and more people contribute, you build up a database. If you want to then do certain analysis, then the data is there. But again, this is individual researcher's choice, how much he or she is willing to share. Then we can do automatic quality control. For example, if the data is of low quality, the system quality control can automatically flag this data as being low quality. For example, too much motion or other quality correction. And we can use application of correction methods, what I have shown, for example, where we can correct for motion and then in anatomical data, then again, the person has then access to the original data that's never lost, but then corrected data might be also available. And we can do data processing pipelining. MR data is analysis intensive, but we can standardize some pipelines, and then this means two researchers will get the same result using the same data. For example, this can be applied for diffusion data. You acquire diffusion data, FA, MD, or if you use NODI, all these things can be automatized, and people do not have to do the analysis themselves. Obviously, they can do themselves, but if they don't want, or if they don't have the expertise or personnel, they can use the automatic pipelines. The last topic is the so-called gadgetron. Some of the sequence I have mentioned, like the wave Kaipi, we have to also develop our spiral. We have to develop also our own image reconstruction. And so Siemens image reconstruction might not be able to do it. Right now, most people do it offline. However, the gadgetron system actually allows to bypass the image reconstruction and do your own image reconstruction and feed it back to the MR scanner host computer, and which then means you can have, for example, your own correction mechanism, your own image reconstruction algorithm in MATLAB or Python. You can, with the gadgetron, you can bypass the MR host system, but the data can be fed. So for the user is not even aware that something's happening. The user only sees the reconstructed data and so on. With that, I'm finishing. So just to give a review what I've talked today, I know I have covered a lot of topics, and it was not a standard talk, but I hope I could illustrate to you what my group is currently working. So we work a lot on MR physics. I'm very much interested in cognitive neuroscience. At this moment, we do a lot of computational approaches, but I am also very experimentally oriented. And then we do clinical neuroscience at this moment, a lot of dynamic susceptibility contrast, but I'm also interested in diffusion and anatomical imaging. And then lastly, I presented our data management system we are currently establishing at the Prisma at the Krembil Brain Institute. But we are also happy to talk with you about sharing, and if you're interested, then we can talk about it. And with that, I want to thank again my group and you for your attention.